Hey guys, I hope everyone had a great Thanksgiving and you got some rest and you did some shopping and you enjoyed your little break and you're coming back ready to push hard until the end of the year. So, this is going to be our first series in our 3-part body systems. Remember I told you the AP exam and college board really only focuses on nervous, endocrine, and immune system. So those are the only ones that we're really going to be doing in any kind of depth. So we're going to start off with the nervous system because it's my favorite and it's really cool and I hope you enjoy it. Okay, so the whole reason that as an organism you have a nervous system is so that you can respond. The faster you respond, the more accurately you respond, and the quicker you are able to respond again, the longer you tend to live. Now, we don't really see that for humans so much, but we definitely see that in the wild. When we look at our predator and prey relationships, prey who exist or live longer and don't get captured and eaten tend to be the ones that can run faster and think smarter and can analyze a situation and change what they're doing as a response to that situation. That's one of the main reasons you need one. It helps you to survive. So what is your nervous system made of? It's really a huge part of your body, but it exists as just a couple of cells. The most important ones would be those dendritic cells called neurons. So this is what's pictured here. This whole thing would be a neuron. Now, neurons are just regular cells, but they look very different to the cells that you've become accustomed to seeing. It is a type of animal cell, but it's an animal cell that has one very long, stretched out section that's filled with mostly just cytoplasm, and then all of the organelles are held in this region, which is called the cell body. That long, stretched out region is called an axon, and really all that it's done is it's an elongated section of cell membrane and cytoplasm. But what that axon does is it acts as a conduit. It acts as a transfer system. So literally the electrical messages that your nervous system sends all run along or pass along this axon. So if you look at this whole picture, you'd see how we have one neuron right here, and then we have another neuron right there, and that's how the message goes. It goes from one dendritic region all the way through the axon to another dendritic region, all the way through the axon, and it just keeps passing and passing and passing. You know that I always say in biology that structure and function go hand in hand. So if you look at this cell body region of your neuron, some people also call it a dendrite, you notice how it has all of these little dendritic, they almost look like roots and root hairs. Pretty much what they are, they're entry points for signals. So this one cell can interpret multiple signals passing along these little dendrites. Okay, and then they all get focused in the cell body, and then they get transferred on as they need to. Now, even though we can access a lot of different stimuli, there's only one path for that stimulus to leave through and go on to something else. So we have a lot of entry points, but we only have one path out. The dendrite is designed the way it is. It's stretched out like that so that it transmits signals from one cell to another, to another, to another. Excuse me. So this is the cartoon version, and up here is what the real cell looks like. It's pretty much the same thing. Now, we kind of talked about this a little bit when we talked about cell communication. There is a small gap, a small space between one dendrite cell and another dendrite cell. Unfortunately, as I told you before, electricity can't jump spaces. So when we get to that small section called a synapse, we're going to have to change the method in which we transfer our information. And that happens in the form of a neurotransmitter. We'll talk more about it later on. All right, so neurons are really cool. So here are some fun facts about them. They're the most specialized cell that you have in your entire body. They're also one of those cells that you tend not to replace very often. And they're also the longest cell. Like, it is the longest cell in every living thing's body. If you're a blue whale, that means that your neurons can go anywhere between 10 and 30 meters. If you're a giraffe, it means that your neurons can go anywhere between like 4 and 5 meters. And even in humans, as little as we are compared to a blue whale or to a giraffe, our neurons can be as long as 1 to 2 meters. All right? Because they're so long and because there's so many of them and because of the ways that they're connected, they allow our responses to be very, very quick and sharp. All right, so how does this whole transmission of a signal thing work? I want you to think of when you were little and you played with your mom or dad's or uncle's dominoes. If you were like me, I like to line them up and try to knock them down so that they would all knock, one would knock the other, until they all went down in a pattern. That's kind of how a signal works. You have to have a signal, you have to have a trigger for the signal, so a start, and then the signal has to propagate, it has to keep traveling, and then once it's gotten to the end, the whole thing needs to reset itself. So if this were dominoes, the signal would be your finger pushing that first domino. The propagation would be each domino in front of the other pushing on the one behind it until they all fell down. The resetting would be when you went on your hands and knees and picked every domino up and put it back in place so that you could do it all over again. That's the same premise that a neuron works based off of. All right. So in our neurons, we don't have dominoes, of course, but it's still a similar concept. The first thing that we do have are a bunch of protein channels that are all set up along that plasma membrane that stretches out on our neuron. When one is opened, it causes the rest of them to open in succession. So this is what we call an all or nothing response. Either the neuron opens, either the protein channel opens and causes all the other protein channels to open, or the protein channel doesn't open and none of the others open. So it's all or nothing. Either we all open or none of us open at all. So as that first channel is open and it changes the environment just a little bit, the next channel opens and the next and the next and the next, so that we have this wave-like action that travels along our neurons, sending that signal. Now, as the signal passes, the proteins that have already been utilized reset so that neurons can react again. Okay? And we're about to see exactly what I mean by that. All right, so you know that your body exists in an aqueous environment. You're about 75% water, and then about 3% of that, or in that, are these dissolved ions like chlorine and potassium and sodium. And a lot of people tend to forget them, but this is where they come into play, and this is why it's so important that your body doesn't ever run out of its ions. When you start having ion deficiencies, that's when the doctors start prescribing things like, oh, you need to drink more Gatorade, or you need to drink salted water, because you're trying to get that ion balance, that electrolyte balance, back up to where it needs to be. So that being said, your cells are surrounded in liquid and ions. And you know that there are two kinds. You should know this from chemistry. Anions will be your negative ions, like chlorine, for example, whereas cations will be your positive ions, like potassium, for example. Now, the anions tend to be more concentrated inside of the cell, meaning there's just a lot more of them. Excuse me if my voice sounds weird. Just kind of woke up. But anyway, which means that there tends to be a lot more of them inside of the cell than there are outside of the cell. The most common kinds are going to be chlorine ions, which are definitely found in great amounts in your cell, and also a lot of our negatively charged amino acids. Excuse me. Now, your cations kind of do the opposite. They're more concentrated in the extracellular fluid, so the fluid outside of the cell, so the fluid that's bathing the cell. And mostly what you'll see would be sodiums that tend to be on the outside. You'll have some potassium scattered as well, but it's mostly sodiums. Now, these channels work in multiple directions, but they actually have a condition where they tend to leak potassium ions. So potassiums that are found inside of the cell over time will eventually just diffuse through that protein channel and end up outside of the cell. Well, if you look at the picture here, you're going to realize that that creates a very negative environment in your cell, and it's going to create an even more positive environment outside of your cell. So, important point to take away from this picture. Because of the way that ions behave and because of the way these channels behave specifically, we're going to have a negative environment inside of our neuron and a positive environment outside of our neuron. Excuse me. So this creates voltage because we have two opposite charges on opposite sides of our cell membrane. Because don't forget, this is a cell neuron. So it has two sets of plasma membrane, if you want to think of it that way. You're going to have one charge situation going on at the top part of the cell, and that same charge situation is going to be duplicated at the bottom part of the cell. But always the inside of the cell is going to be negative, whereas the outsides are the parts that are positive. So this creates voltage, and it makes your membrane polarized. Polarized just means, again, we're negative inside, positive outside. We have a charge gradient. It's kind of like a concentration gradient, but this time we're dealing with charges. Okay? This allows your neuron to kind of store energy, so it's almost behaving like an Energizer battery or a Duracell battery at this stage. All right? So, what does this mean? Why is this voltage important? Well, we've been able to measure the voltage in neurons that are stimulated, meaning they're active, and neurons that are unstimulated, meaning they're resting. When a neuron is not being utilized, when no message is being sent and that neuron is unstimulated, it is at what we call the resting potential. So I'm going to say that again so you can write it down. The resting potential is the voltage of a neuron that is unstimulated. It is not passing on or transmitting a message. So again, the resting potential is the voltage of a neuron that is not stimulated. It is not passing on any kind of signals. And there's an actual number that goes with that. The resting potential for an unstimulated neuron is negative 70 millivolts. So, we know we have resting potentials. We know we have protein channels. How are we really going to send this message, this signal, this impulse? Well, we need a stimulus. The stimulus means that our nerve is triggered, our nerve is activated somehow. So we're no longer at resting potential. Our voltage has changed. That's what a stimulus does. And a stimulus can come in multiple forms. It could be pain. It could be the changing of temperature, like, going from hot to cold or cold to hot. It could be another hormone or neurotransmitter triggering that particular nerve. So the stimulus just means that we've activated our nerve. We're no longer at resting potential. When a nerve is triggered, it goes from resting potential to what we call threshold potential. So think of it as we've just knocked over our first domino. At threshold potential, two things happen. The sodium channels in the cell membrane are going to open up. When they open up, natural order is going to take place, and sodium ions are going to diffuse through that channel into the cell. Now here's where you have to start thinking. You know that sodium is a cation, and I told you the outside of the neuron is positive and the inside is negative. If we start pushing sodiums into our neuron, we're going to change the charge on the inside. Okay? So if you look at the picture, as those sodium ions start diffusing into the cell membrane, we're going to take this internal charge from being negative and it becomes positive. So our charges reverse wherever our sodium channels open up. All right? So when we are at resting potential, we are negative inside, positive outside. Once our nerve has been triggered and we start getting to threshold potential, we're going to have a reverse of charges. Instead, slowly over time, we're going to become positive on the inside and negative on the outside. This is called cell depolarization. So the cell is becoming depolarized. We are switching where our charges are normally found. So excuse me. When a cell is depolarized, it becomes positive on the inside, hopefully you're writing this down, and negative on the outside. Let's say it again. When a cell becomes depolarized, or through cellular depolarization, the cell becomes positive on the inside, right here, and negative on the outside. All right, so we know how the impulse starts. What we just went through about how we start switching those charges, that's how the impulse starts. But how does it propagate? How does it continue? Well, the impulse travels down the neuron almost in the form of a wave. Because when one protein channel opens, it causes the next and the next and the next to open. When we talked about cell communication, we talked about ion-gated channels. Another version of an ion-gated channel is called a voltage-gated channel. It works in the same way, but this time the channel is going to open or close based on the charge. Okay, so when that first protein channel opens up and causes that sodium to diffuse in, the next one is going to do the same thing and sodium will diffuse, and the next one and sodium, and the next one and sodium, until we start literally going from negatives to positives all the way along this neuron. Okay, now because we're changing the charge, we're going to be changing the overall voltage as well. Don't forget, charge and voltage go hand in hand. And this is called an action potential. So remember my domino example? Think of this part of the think of this part as the rest of those dominoes falling. One domino pushes the other, which pushes the other, until they all kind of fall down. That's what's happening here. This channel opened up, caused sodium to diffuse through, which triggered this channel to open up and cause sodium to diffuse through, which will trigger the next one to do it, and the next, and the next, and the next. All right, so that's the first part of how our impulse is going to travel. But we kind of have to reset these channels, because if they're all open and sodium has completely diffused through and nothing causes our charges to reverse, then it's almost like this neuron has locked itself. This neuron won't be able to be used again until it's reset. And this is how the resetting takes place. Potassium channels open. And when the potassium channels open, they're going to cause potassium ions to diffuse out of the cell. Now remember, we just put sodium in. Sodium's positive. Now we're about to start losing potassium. Potassium's also positive. So we're taking some of the positive ions out of our cell, which would cause our internal charge to go back to being negative. Now here's the deal. The potassium channels will open, but they open a whole lot more slowly than the sodium channels. This ensures that the message, that impulse, that signal is being transmitted all the way down before we interrupt it. If we started changing the environment before the message passed, the message would stop. So that's one of the reasons that the potassium channels open slower. It is to ensure that the message gets carried all the way through before we reset the neuron so that it can be utilized again. So when the neuron gets reset, our charges get reversed. We go back to being negative on the inside and positive on the outside. This part would be like you setting your dominoes back up. That is your reset. Okay? Now, just like how the sodium diffused in and caused a wave of positives to come in and change along this neuron, when these potassiums start diffusing out, it's going to cause the negatives to come back in another wave, because this potassium pump opens and potassium leaks out and it becomes negative, and then the next and the next and the next and the next and the next. So it goes on in this wave-like motion. So the way I like to think of it, I always envision two combined waves traveling down a neuron. Your first set of waves is going to open up your ion channels, which is going to cause your signal to move in only one direction. Okay, so from the cell body all the way through the axon to the dendrites of the next cell, into its cell body, all the way through its axon, and it just keeps going. When we start resetting those channels, when we start pushing or diffusing potassium out of the cell, it stops the activation of our sodium channels. So it prevents the signal from moving in the wrong direction. So if you would envision with me, we want our signal to travel this way. I need to make that pen bigger. Hold on. We want our sodium to travel in a very specific direction. Excuse me. All right. So we want it to travel this way. But if we have sodium channels opening here and here all over the place, then the wave could just as easily travel in the opposite direction. And that doesn't do us any good, because we're sending the signal back from where we got it from. That doesn't help us to run faster or to hide longer or do whatever it is we need to do to survive. So we want our signal to be propagated in one direction and one direction only. Having this potassium channel that delays its opening ensures that that happens. It ensures that the signal has passed before we reset those sodiums, allowing them to take another signal in potentially the wrong direction. Okay, so this potassium pump is almost a fail-safe. All right, so your action potential is going to propagate as that signal passes. Happens in a wave, also known as a nerve impulse or an action potential, same thing. And it's quick. It's very, very quick. Like I'm telling you that the potassium delays, and it does, but we are still able to send these signals in milliseconds. So let's say you put your fingers on a hot stove or a hot plate. Even before you start to feel the burn of, the heat from that stove blistering those cells in your fingers, you've already pulled your hand back. That message literally went from the pain sensor in your body, sending a signal to your brain saying, hey, this doesn't feel very good, maybe we should move our hand. Your brain then in turn has to interpret that message and then start firing off your neurons, stimulating them to send a message to the muscles in your arms, causing them to contract, thereby pulling your hand away from that hot stimulus. But when we do it, it doesn't seem like it takes that long, because literally you touch it, you pull your hand back. So these messages travel lightning speed. Okay, so let's talk about these voltage-gated channels a little bit. They're ion channels that open and close in response to changes in the charge across your membrane. So as the charge changes, the ion channels either open or they close. Your sodium channels will open quickly, and they do it in response to depolarization. So they do it when the charges get switched. Now they open quickly, but they close slowly. Again, that ensures that the signal keeps traveling in the direction it's supposed to. Potassium channels open slowly, again as a fail-safe to make sure that the signal keeps on traveling in the direction you want it to. And they also do that in response to depolarization. When we went from a negative environment to a positive environment, it is going to cause your sodium channels and your potassium channels to open. It's just that your sodium channels open really fast and your potassium channels open a whole lot slower. Okay? So again, that's the whole structure and function thing. All right, so how does it reset? It reset, no, we've already talked about this a little bit. So your neuron has been fired, it's been utilized, it sent its signal, but if it stays in its current situation, the neuron is locked. It can't send another signal. So the whole thing needs to be completely reset. All the parts and pieces need to go back to the way they were before that first sodium channel opened. So the first thing that needs to happen is your sodium needs to move out of the cell, and this allows the inside of the cell to go back to being negative. Then your potassium needs to move back into the cell, and this allows the outside of your cell to remain positive. These are going to be moving against concentration gradients, because if you look at the picture, you'd realize where we have a greater concentration of what. So that means we're going to have to utilize ATP, we're going to have to use active transport, we need some kind of pump. That pump is going to be a sodium-potassium pump. It can transport both molecules in opposite directions, exactly where they need to go. And this is the real resetting of the neuron. So when the sodium initially diffuses, you're just preventing the signal from coming back. When we do all of this, when your sodiums move out and your potassiums move in, this is where you're truly resetting up each and every single domino, spacing them properly and getting them ready to fall down again when you push the first one. So this is what a sodium-potassium pump looks like. Like I told you, it is capable of carrying multiple ions at the same time, and it carries them in 2 different directions. It is going to utilize ATP because we are doing this against a concentration gradient. So the way it works is through active transport. We're utilizing that ATP. We're getting that energy so we can move our molecules where they need to go. And it works by a certain ratio. The pump is going to transport or to pump 3 sodium ions out and 2 potassium ions in. So for every 3 sodium molecules it moves, it also moves 2 potassium ions. And it's through this pumping of, it's through this movement of this pump and this pushing of these ions in and out of the cell respectively that the membrane gets reset. Obviously, if we're using ATP, it means we're going to need some kind of molecule to bring that energy back in, normally in the form of a carbohydrate. Okay, so once we've gone through the whole process of utilizing the pump, we're back at our scenario where the outside is very, very positive, the inside is only slightly positive, and the inside is very, very negative. Look at the concentration gradients. Lots of positives here and here, so outside is more positive than inside. No negatives, so inside is definitely more negative than outside. So at this stage, we are right back at our resting potential. We're back at that negative 70 millivolts. We're getting ready to be fired, to go up to threshold potential, to perform an action potential so that a signal can be transmitted. So, of course, you know there's going to have to be some graphing or some math involved. So can we graph this? Yes, we can. This is called an action potential graph, and it takes you through every single step from our resting potential all the way back to the resetting of that neuron. So, part 1, if you follow me, this is part 1 right here. Look, we are at exactly negative 70 millivolts. So that is going to be our resting potential. Nothing's happened yet. The neuron isn't stimulated. Nothing has triggered the neuron. We're just waiting for some kind of message to come in. Now, part 2 is where we get that initial stimulus, and we're working our way up to threshold potential. Threshold potential happens at around negative 55 millivolts. You might want to write that down. Okay, so threshold potential, which pretty much signals the start of depolarization, of those channels opening up, is reached at negative 55 millivolts. So from this point, from that negative 55 all the way up to about negative 30 millivolts right here, so this line that my laser is showing you right here, that is depolarization. In depolarization, we're going to have that switching of charges, So we're going to go from what was once a negative environment on the inside to a positive environment on the inside. So during depolarization, those sodium channels open up, the potassium channels are closed, and sodium rushes into the cell, thereby creating that positive environment. Excuse me. Now, once all of the sodium channels have opened and the signal has been passed on, the signal has been propagated the way it should, those sodium channels are going to close. And instead, the potassium channels are going to open up. This happens at 30 millivolts, so right at this apex region of your graph right here, and it triggers repolarization. In repolarization, we're going to reset our charge. So we were negative inside of the cell. Because sodium came in, it made the inside of the cell positive. Now we're going to pump potassium out to make the inside of the cell negative again. So we go from negative to positive to negative. And that's what repolarization is, and it happens right in this region here. So we're resetting our charge gradient. Now remember, I told you that the potassium channels, they close fairly slowly. They stay open for a fair amount of time. That causes what we call an undershoot. So we get to the point where the cell pretty much loses all of its positive charge. And that's what our undershoot is. It's this region right here. So it kind of corresponds to threshold. And then we gently ease ourselves back up to resting potential. So undershooting happens at around negative 75 millivolts in that vicinity. Okay? And it happens because we have lost too many positive charges, because those sodium channels close so slowly. Another name for the undershoot is hyperpolarization. All right? And this is an action potential graph. Excuse me, guys. I'm so sorry. So, let's talk about the structure of these neurons a little bit more in depth so that we can figure out how they work as well as they do. So, your axon is coded by what we call a Schwann cell. Yes, it was named for the man who discovered them. And those Schwann cells wrap themselves around your axons. If you look at your picture, this cartoon version, this yellow section here and here and here, that's the actual axon. All of this white stuff that looks like a carpet that you've rolled up or a piece of paper that you've rolled up, excuse me, those are your Schwann cells. This entire thing is a Schwann cell. Look at how the nucleus pokes out right there. So it literally wraps up the axon, insulating the axon. This helps to speed up the signal, because see how the axon is exposed at certain regions? Like if you look at this part of the picture, you have a little bit of axon here, and then there, and there, and there, all the way down. Well, when the signal travels along the axon, what it really does is it jumps over the Schwann cells. So it jumps from axon to axon to axon to axon. Does that make sense, hopefully? Know why I'm asking you questions. It's not like you can answer me. But anyway, if you don't get it, let me know in class. So literally the signal is going to jump from this point to that point to that point to that point instead of traveling all the way through. It helps the signal to move faster. So we call it hopping from node to node. Each node, by the way, has a name. It's called a node of Ranvier. Again, named for the person who figured it out or discovered it. And this is what we call saltatory conduction. If you've taken French, salt means to jump. So, saltatory conduction. Ranvier obviously was French, so was Schwann. So, what does that mean for us? Well, it means that we can transmit a signal in about 150 meters per second versus being able to transmit a signal 5 meters per second. Speed distance, we're talking 330 miles per hour versus 11 miles per hour. So by jumping, we can conduct a signal 150 meters per second. If we were to travel through every single bit of that axon, that same signal would be transmitted at 11 miles per hour or 5 meters per second. That's the major difference. All right. Now, I don't know if you have any family members with this condition. One of my very favorite aunts in the whole wide world has this condition. It's called MS, or multiple sclerosis. And it is a condition that specifically affects your neurons. Specifically, it affects the myelin sheath, those Schwann cells. And it's what happens when your immune system, specifically your T cells... Alright guys, I know I sound like I'm dying. I promise you I wasn't sick this whole time. I decided to make this video for this morning and for you guys this morning, and now I'm dying. Oh well. So, I was saying, in multiple sclerosis, it's what happens when your immune system, your T cells specifically, no longer recognize the myelin sheaths, those Schwann cells that surround the axon, and they start attacking them and breaking them down, and this causes a loss of signal. So it's not so much that the signal itself gets lost, but the signal travels slower, because the way saltatory conduction works is everywhere there is an exposed axon node, the signal goes to that point. In multiple sclerosis, I'm going to kind of draw over this, hopefully it works. In multiple sclerosis, we break down this myelin sheath so that the axon is exposed. And now instead of going from this point to this point, the signal now jumps from here to here, and then from here to here, and wherever else it's broken down. So it slows down the message. Alright, we'll watch this video in class. So, what happens at the end of the axon? We know that the axons aren't actually connected to each other. There is a small space between one axon and another axon. And we also know that electricity can't jump into thin air. So, how does the signal pass from axon A to axon B? Well, you'll see. So, we used to be an electrical signal because neurons transmit electrical signals. But we have this small problem where we have this space between the end of one neuron and the beginning of another neuron. And the electrical signal cannot jump through or pass through that dead air. So how are we going to keep the signal going? Well, it's simple. We change from an electrical signal to a chemical signal. Duh. Sorry, I couldn't resist that. I just felt like I had to add that in. Anyway, so here's what happens. So your action potential depolarizes the membrane when you get to the end of your current neuron. Now, the term that we use the terms presynaptic cell or neuron and postsynaptic cell or neuron. So we do everything based on where that synapse is. The synapse, by the way, is the actual space. So this right here, that is your synapse. Let me take that off so you can see the rest of the stuff that I need to show you. So this part, sorry, hold on. Here we go. This part of your neuron belongs to the presynaptic cell. This is where the signal is coming from. This part of the neuron belongs to the postsynaptic cell. This is where the message needs to continue. This is where the signal is going to continue once we get through this little spatial issue that we have here. So this is how we get through that space. Your action potential reaches the end of the axon. When it does that, it depolarizes the membrane. Don't forget, I know depolarizes sounds like the scary word. It just means that we switch the charges. So we were negative on the inside and now we're positive on the inside. That depolarization is going to open up a different kind of channel, this time a calcium channel. And hopefully you remember that calciums are cations. So these calcium channels get opened up and calcium molecules start coming into the end of your presynaptic cell. This is going to trigger neurotransmitter vesicles. So these little round things, those are the vesicles, and the little red things are the neurotransmitter. This particular neurotransmitter is called acetylcholine. So the neurotransmitter is made and it's put in these vesicles and it's just kind of sitting there waiting. Okay? When the calcium channels open up, it causes these neurotransmitters to move to the membrane and to fuse with the membrane. When they fuse with the membrane, the vesicle opens up and the acetylcholine, the neurotransmitter, is going to diffuse out of that presynaptic cell. So it diffuses through the space. It is now carrying the message, by the way, and it is going to attach itself to ligand-gated ion channels. You know how these ion channels work. It's the same kinds that we talked about when we did cell communication. So in this case, our neurotransmitter starts acting like a protein, like a signal molecule, like a ligand. So it's going to bind to a protein receptor. It's going to cause the ligand-gated channels to open up, and the neurotransmitter then is going to get broken down and reabsorbed. When these ligand-gated channels open up, guess what's going to happen? Sodium molecules are going to diffuse in, potassium molecules are going to start to diffuse out. Depolarization happens, but now it's happening on the postsynaptic cell, so the message just got passed. Okay, so let's summarize. So our message is being passed on in our presynaptic cell. It gets to the end of that presynaptic cell, and the action potential, what's carrying the message, depolarizes the membrane at the end of that presynaptic cell. Because that membrane becomes depolarized, again, depolarized just means that now we're positive on the inside. It causes calcium channels at the end of the membrane to open up and calcium diffuses in. Neurotransmitter, which is now encased in a vesicle, because of these calcium ions coming in, is going to move to the end of the membrane and fuse with the membrane. By fusing with the membrane, the vesicle opens up and the neurotransmitter, which is now responsible for getting the message to be continued, is going to diffuse through the synaptic cleft, okay, which is that space between the two membranes. When this neurotransmitter diffuses through, it is going to act like a ligand, and it is going to bind to a ligand-gated channel, which is a protein receptor. By binding, it causes these channels to open up. When these channels open up, it causes sodium ions to move into the postsynaptic cell, which causes this membrane to depolarize, thereby transferring the message on. Okay, so once we get to the postsynaptic neuron, we know that our impulse it's still going to be triggered. It now comes in the form of a chemical signal, like I said, which is opening up these channels. So right here, this molecule right here, that is the acetylcholine. it is going to bind, it is going to cause the ion channels to open up, and there goes sodium in, and there goes potassium out. Okay? This is going to allow us to depolarize this entire membrane. We're just making the inside of the membrane positive, thereby propagating the signal. And that's how a signal works. That's how a signal gets transferred. It's all about the sodium and the potassium. So, at our postsynaptic membrane, we have what we call postsynaptic potentials. And they fall into two categories: excitatory postsynaptic potentials, or EPSPs, and inhibitory postsynaptic potentials, or IPSPs. EPSPs are depolarizations that bring our membrane potential closer to threshold. Once we get to threshold, it means that we can actually go through depolarization and then repolarization, and then we can reset. So these potentials will cause a signal to be passed on. They propagate a signal. Inhibitory postsynaptic potentials are hyperpolarizations that take our membrane away from threshold. So we're not getting close excuse me, to that -55, we're not going to open up our sodium channels. Pretty much what IPSPs do is they stop the signal. Let me write that in for you. So EPSPs continue the signal. Whereas IPSPs stop the signal. See, I've gotten so much better at writing with this pen. Very proud of myself. Alright, so this is another video that shows exactly how the synapse works, how those neurotransmitters come in and all of that. We'll watch this in class because this particular app doesn't allow me to take the videos that I used to have in my PowerPoints and show them in this video. So we'll just have to watch it in class. Someone remind me. So right here, all of these are just types of neurotransmitters. Whoops, sorry, hold on. Yikes. Tried to hit my laser function, didn't really work. So again, these are just different types of neurotransmitters and what they do. Acetylcholine is the one that we always use as our example, and specifically what it does is it transmits signals to skeletal muscle. So remember that example I gave you earlier where you touch something hot and your body makes you pull your hand back? Well, acetylcholine is going to be the neurotransmitter that goes to your muscle, your skeletal muscle attached to your bones, telling those muscles to contract or to relax. Two other good examples would be epinephrine, also known as adrenaline, and norepinephrine. And these two control our fight or flight response, which we kind of talked about a little bit. Then there's dopamine, which is found pretty much everywhere in your body, especially in your brain. It affects your sleep, your mood, your attention, your learning. When there isn't a lot of dopamine in your brain like there should be, it's associated with Parkinson's disease. And when you have way more dopamine than you really need to have, it's linked to schizophrenia. So dopamine is one of those neurotransmitters that's highly important, but you need just the right amount. Too much, and you're schizophrenic. Too little, and you shake. You have Parkinson's disease. It's not just shaking, but that's one of the classic symptoms. Then there's serotonin, which is also widespread in the brain, and it affects your sleep, your mood, your attention, and your learning. Notice how both of these affect sleep, mood, attention, learning. Guess how this works? You don't sleep well, you're not in a good mood. You're not in a good mood and you're tired, you don't have a good attention span. You don't have a good attention span, you can't learn my AP biology. So Josh and all those other people that like to go to bed at 3:45 in the morning, yeah, important. Sorry, couldn't help it. All right, so your neurotransmitters are kind of the weak link in your nervous system. For the most part, the nervous system works really, really well, and it's almost foolproof. But if something were to attack the nervous system or could attack the nervous system, that's where its weak point would be. in the form of these neurotransmitters. But the neurotransmitters can't be helped because your nervous system deals in electricity, and because of those spaces, electricity can't move from one cell to another. So you need the neurotransmitters. The reason it's a weak point in your nervous system is because any substance that affects either making or the action of those neurotransmitters, or even mimics them in terms of the nerves that they can stimulate, that's going to affect the whole workings of your system. So that's why some of our gases, like nitrous oxide and carbon monoxide, that's why they're deadly, because they mimic neurotransmitters that at this point in time you don't need them to mimic. That's why some types of drugs, like the stimulant drugs and the depressant drugs like caffeine and nicotine, are very, very mood altering, because they mimic dopamine and serotonin. All right, our hallucinogenic drugs do the same thing, right? Like LSD and all that stuff. So, not good, kids, not good. So let's look at an example of how a neurotransmitter can be a weak point for your nervous system. There is an enzyme called acetylcholine esterase, and it is an enzyme that breaks down and recycles the acetylcholine neurotransmitter. And we have it in great amounts in our bodies because instead of remaking acetylcholine every single time we need to send a signal, your body just kind of utilizes what it already has. Well, neurotoxins like snake venom and some insecticides, for example, block the active site of the enzyme acetylcholine esterase. If acetylcholine esterase can't break down acetylcholine, it means that when a new signal comes in, there is no acetylcholine to take the electrical signal and make it a chemical signal, thereby propagating it on the other neuron. There's no way that the signal can continue because there's nothing to take the signal there. All right? It's like if your mom says, call her, but your phone's dead, you can't call her. Same situation here. So, whole system shut down because we don't have the enzyme that we need to recycle the neurotransmitter that we need. So when poisonous snakes that have neurotoxin venom bite you, this is what happens. Okay? Your messages can't be sent, and then your body doesn't know what to do. Questions to ponder. I started putting these in recently. Why are axons so long? Why have synapses at all? Why do we need to have this space if it's creating such an issue? How do mind-altering drugs work, like caffeine, alcohol, nicotine, marijuana? Do plants have a nervous system? And do they need one? We'll talk about it in class. All right, so you can take your nervous, and if you're in anatomy, when you guys do the nervous system, this is the part of the system that you focus on more than anything else. But animals especially have a complex nervous system that's actually made up of two parts: the CNS, or central nervous system, and the PNS, or peripheral nervous system. The central nervous system is where integration and interpretation takes place, and it only includes two parts: your brain and your nerve cord. The peripheral nervous, or spinal cord, nerve cord, spinal cord, same thing. The peripheral nervous system, or PNS, is the part that's doing the carrying and the bringing in of information. So your PNS collects information and takes it either into the CNS or out of the CNS. It consists of the neurons and the nerves. So, two parts to the nervous system of an animal: CNS, which is the brain and the nerve cord, and PNS, which is the neurons and the nerves. The CNS interprets information that's brought in. The PNS brings information into and out of the CNS. All right, so this is what it looks like. So here and here, that would be your central nervous system. Everything else would be your peripheral nervous system. That's all that this picture shows. When I was in college and I had to teach anatomy, My kids were responsible not just for knowing the general basics, they also had to know pretty much all of the nerves, what they went to and what they did. Have fun. I'm kidding, you don't have to know that. All right, so here's how it works in terms of processing. You have two kinds of receptors or neurons. You have two kinds of neurons, afferent neurons and efferent neurons. Afferent neurons bring information in, and efferent neurons bring information out. So let me say that again. Two kinds of neurons, afferent neurons and efferent neurons, and they sound very similar if you're talking quickly. Afferent neurons bring information to the CNS, and efferent neurons bring information out of the CNS. So this is bringing information in to be processed. This is the decision that's been made, and now we're taking that decision and executing it. Your system, excuse me, is also going to need receptors to do and gather the information. So that's these sensory receptors right here. These sensory receptors get triggered by internal and external stimuli. So that could be a raise in blood sugar level. It could be a hot plate. Someone has stabbed you with a knife. It could be any kind of stimulus. These sensory receptors are going to take that information, send it through afferent neurons to the CNS where it's going to be processed. After a decision has been made, after we've decided how are we going to deal with the knife that's now sticking out of your hand, we're going to take that information, that decision, and we're going to send it to the body in the form of, or through, an efferent neuron. Now, those decisions can have one of two destinations. They can go to your motor system. Your motor system is pretty much all of your muscle, all of your skeletal muscle. And it might say, hey, take that knife and pull it out of your hand, or move your hand away from that hot plate, or whatever it is that's going on. Or the message can go to your autonomic nervous system. Your autonomic nervous system controls all of the things that we have to do every single second of every single day to stay alive, but we don't ever think of, like breathing, for example, and the beating of your heart, and the contraction of the smooth muscles inside of your blood vessels that help your blood to keep pumping. All of these things are controlled by your brain, and they have to be stimulated, but you don't have to think in order to do it, if that makes sense. Like, I don't sit here and I go, okay, heart, beat, 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 beat now, beat now. Okay, stop beating. No, I don't do that. My heart just beats because the autonomic nervous system, or ANS, controls that. It does it for me. All right. The autonomic nervous system is divided into three sections: the sympathetic division, the parasympathetic division, and the enteric division. You don't really need to know what specifically those three parts are in charge of. Just know that collectively they control smooth muscle, cardiac muscle, and glandular stimulation. All right. So how does it work? This is just another diagram that you might potentially see. Your nervous system works hand in hand with your five senses. All right. So one of your sense organs, eyes, nose, skin, whatever it is, gets stimulated. That is what we call sensory input. So your senses are putting information into your brain. That message is going to travel to your brain. Your brain is going to interpret the message, and then it's going to send a signal causing you to do something. Okay? That signal is going... Hold on a second, guys. Sorry. So like I was saying, the motor output is going to cause some kind of reaction. The muscle that motor output stimulates to cause you to do whatever it is you're going to do is called the effector muscle. So you see a ball coming to you because you're playing soccer. Your brain interprets how fast the ball is coming, what angle the ball is coming at, and what's the best thing for you to do in response to that ball. That decision gets sent out to your leg muscles, telling you how to kick the ball, or if to stop the ball, or whatever it is that you're supposed to be doing. All right? And of course, this is the brain where it all happens. you'd notice that your brain is made up of lots of parts and pieces, and they're all in charge of different things. You don't have to know these specifically. I just thought this was cool, just to show you how it works. All right, so that's it. Hopefully it wasn't too painful. And as usual, if you have any questions, first 20 minutes of class when I see you guys on Monday and Tuesday will be dedicated to answering those questions and going over concepts that you struggled with. Enjoy the rest of your very short weekend, and I'll see you soon. Bye, guys.